Hello, my name is Jinyu Wu, and my study is the effect of a high glucose diet on amyloid toxicity using CNI-143 as a novel treatment for Alzheimer's disease with the simulation of intracellular A-beta aggregation. Alzheimer's disease is the sixth leading cause of death in the United States. Currently, 5.4 million Americans are affected by Alzheimer's disease, and that number is expected to increase to 16 million by 2050. In 2013, over $220 billion were spent on Alzheimer's disease. Alzheimer's disease is the most common form of dementia. Alzheimer's disease occurs when there is a buildup of amyloid beta plaques in the brain. First, the amyloid precursor protein APP cleaves by the beta secretase enzyme into amyloid beta monomers. These monomers are then either degraded or joined into dimers. Next, the mechanism behind Alzheimer's occurs. The dimers combine with the monomers or other dimers to form oligomers. The oligomers then form into insoluble toxic amyloid beta plaques. Soluble amyloid beta is essential to neuron function. However, the toxic plaques can be dissolved by proteasome autophagy.
Neurons drift thousands of molecules of protein. In an Alzheimer's brain, some of them are suspicious protein fragments called beta amyloid. Sticky and resilient, bits of beta amyloid clump together to form an expanding mass called a plaque, which attaches itself like a barnacle to the outside of the neuron. Sensing a hostile invader, the brain's defensive cells fight back, releasing powerful chemicals. But the counterattack backfires. The chemicals set off a series of reactions that slowly destroy the neuron.
Schlotterer 2009 used C. elegans to model glucose toxicity. High glucose was shown to reduce mean lifespan from 18.5 days to 16.5 days, and maximum lifespan from 25.9 days to 23.2 days. The formation of methylglyoxal modified mitochondrial proteins and ROS was significantly increased by high glucose conditions and reduced by mitochondrial uncoupling. Overexpression of the methylglyoxal detoxifying enzyme glyoxylase 1 attenuated the life-shortening effect of glucose by reducing AGE accumulation by 65% and ROS formation by 50%, and restored mean lifespan from 16.5 days to 20.6 days, and maximum lifespan from 23.2 days to 27.7 days. The inhibition of glyoxylase 1 using RNAi further reduced mean lifespan from 16.5 days to 13.9 days. Neves 2008 used virtual cell and partial differential equations to model cyclic AMP concentration within a neuron. In Figure C, the cyclic AMP concentration was highest near the dendrites of the neuron and lowest near the body of the neuron. Thus, the small volumes of the dendrites are more severely affected compared with the larger volumes of the cell body. In Figure E, the two different diameters of the dendrite made a large difference in cyclic AMP concentration. The blue in the 3 micrometer diameter shows a low concentration of cyclic AMP, and the red in the 1 micrometer diameter shows a higher concentration of cyclic AMP. Graph F shows the chemographs of simulation results of the concentration of cyclic AMP with respect to time for varying dendritic diameters. Lastly, G shows concentration of cyclic AMP at 600 seconds, plotted against dendritic diameter and its corresponding surface to volume ratio. Proctor 2012 also used virtual cell to investigate the extracellular amyloid beta plaque formation mechanism. His study provides many useful K-values or reaction rates for the different steps of amyloid plaque development. Graphs A through D represent histograms showing percentage of times that amyloid beta exceeds threshold for normal and AD degradation rates. A and B have a threshold of 4. B shows the amyloid beta degradation rate for a person with Alzheimer's disease. Rarely in graph A was the threshold exceeded. Graph C and D each have a threshold of 1, and graph D shows a slower degradation rate. However, these two graphs had more simulations where the threshold was exceeded. Table 2 and Figure 11 show the reaction rates that I will be using from this study. Figure 11 is a map of the various reactions that occur during plaque buildup. Oh, hi! The purpose of my study is to investigate the effect of high glucose on ROS formation and amyloid plaque buildup using C. elegans as a model organism. Next, CNI-143 will be used as a possible treatment to combat insoluble amyloid beta plaques. Lastly, virtual cell will be used to model the formation of amyloid beta plaques in the neuron and muscle cells. And finally, the CNI-143 effects will also be measured using the virtual cell to see the effect on plaque formation. Procedures in the study include using virtual cell, running Western blots to determine the amyloid levels within the C. elegans body, using GFP to also measure amyloid beta expression. Furthermore, the expression of the genes can also be measured by running a reverse transcriptase PCR. Then a gel electrophoresis will be run to accurately depict gene expression. A sushi cell, which is taken from human tissue, will also be used to study the CNI-143 effect on amyloid beta plaques. Another method that can be used to measure ROS levels is using flow cytometry. In flow cytometry, tissue samples are broken up to single cells and the cells flow through this flow chamber. Then, a small laser beam of bright light hits the cells as they pass through the chamber. The physical properties of the cell are then identified. Next, the light detector processes this light signals and sends information to the computer. The filter then directs the light emitted by the fluorochromes to the color detector. As the cells pass through the laser, the fluorochromes attached to the cell absorb light and then emit a specific color of light depending on the type of fluorochrome. The fluorochromes on the cells act like barcodes on groceries as the cashier passes them over the scanner. Here is the molecular structure for CNI-143, the drug treatment that I will be using. And also, here is the agenda for my study. Here is my bibliography, and thank you for listening to my presentation.